Hi everyone, Dr. Ron Bio again here to discuss the mitotic cell cycle. We're going to put it all together here and go from G1 of interphase all the way through a complete round considering a lot of different factors. Okay, so this video is going to summarize essentially everything in the mitotic cell cycle with a specific focus on the genetic material as we transition from interphase to the specific steps of mitosis. So there's a lot of detail here, so please press pause during the parts you're less familiar with. So if you need to get all the details, if you're taking notes, if you're trying to review for a test, press pause to give yourself a chance to catch up if too much is going on. Okay, so this introduction here, there's a big picture idea here on this first slide. And essentially, with the mitotic cell cycle, we take diploid cells and make more of them. If you don't remember what diploid means, there's a video called Beyond the Vocabulary where I discuss the mitotic cell cycle at a beginning level. So it's called Mitotic Cell Cycle for Beginners. That will help you. And that might be a good idea to go back and start there if the term diploid or haploid is confusing. Okay. If you understand what diploid is, you'll understand here that we have a parent cell that's a diploid cell. It's got a 2N number. And essentially what the mitotic cell cycle does is it takes that parent cell and makes two daughter cells. So you go from being one cell with a 2N value to having two cells that have a 2N value. And if all goes well, there haven't been any significant problems, you come away with two identical daughter cells with the same 2N equals 8. Okay, so let's get into the process here. It all starts with interphase, G1 here in this particular picture. So we're going to follow the path of only one of these daughter cells. So yeah, you make two. This one would go into its own pathway, own G1. We're gonna follow the path of one cell here. Okay. If your 2N is 8, that means you have 8 chromosomes in total in this diploid cell. Note that the genetic material is in a relaxed state, okay? So as we transition here, you see in G1, DNA is relaxed, okay? Essentially, it looks like a ball of yarn. It's relaxed, it's decondensed here. Decondensed is another way of saying relaxed. Okay. The first phase of interphase is called G1, which stands for gap 1. So we got gap 1 here. I tell my students to think of it, though, as growth 1, as this is exactly what's happening inside the cell. The cell has just divided. Cytokinesis has just happened. So the cell, the daughter cell, is a little smaller than the original parent cell. In G1, it grows. It produces more membrane, more cytoplasm, and we begin to make more organelles like this mitochondrion here. Keep in mind everything was just divided, so it starts to grow and make more. You can also see a single centrosome here. We lost the other centrosome to the other daughter, so eventually we're going to have to duplicate another centrosome, make some more centrioles down the road at least. So we have a 2N of 8 here, okay? The DNA is relaxed. So we have 1, 2, 3, 4, 5, 6, 7, 8 chromosomes. We have a pair of number 1s. We have a pair of number 2s, a pair of number 3s, and a pair of number 4s. So this gives us a 2N of 8. Let's head over into S phase and see what happens here. So as we transition from interphase to S phase here, the key event that happens is the duplication of the genetic material. Okay, the S in S phase stands for synthesis. It's the portion of interphase in which you're synthesizing DNA. You're duplicating all the DNA in the nucleus. This means all the chromosomes are being duplicated. I've shown you that here by showing the red genetic material. So it's still really loose and it's in a ball of yarn state, okay? The analogy I like to use in my class is copying homework. Not that any student should ever do that, of course, but if you were to forget to do your homework and you ask a friend to help you out, they would have to let you copy their homework so that both of you have a copy of the homework. It would not make sense for them to give you theirs or to tear their homework in half so that you each have some bit of the homework. In the first example, if they gave you their homework, they wouldn't have their homework, right? And if they tore it in half in the second example, well, then neither of you would have a complete assignment, so you'd both be in trouble. So the cell duplicates the DNA so that there are two complete collections of genetic material, okay? In this picture, I'm going to go ahead and draw the duplicates here in red. So this chromosome gets duplicated, this chromosome gets duplicated, so on and so forth. Those are those characteristic sort of X-shaped chromosomes you see in your textbook, okay? Still, our 2n equals 8, and I'll come back around to that in a moment, okay? So we have all of our DNA duplicated here. Chromosomes are now in the sister chromatid state, okay? So they are duplicates of those chromosomes. We're going to transition from interphase, S phase to G2, okay? Again, G2 means gap 2, but think of it as growing part 2. Here the cell is taking somewhat of a molecular inventory to see if it will be able to proceed to the mitotic phase in order to divide this duplicated DNA. One of the things it needs is energy. So the organelles are duplicated, especially that mitochondrion again. We're showing mitochondria being duplicated here. And you also need to ensure as a cell that you have two centrosomes by the end of G2. You need two centrosomes to effectively separate duplicated DNA in mitosis. So that's a key right there. Nothing much has changed from the DNA's perspective as we transition from S phase over here to G2. The genetic material is still loose. So you see that hasn't changed from these two pictures. And it's still duplicated. You can see here the sister chromatids. We've made notes right here to show you that sister chromatids are red and black, and the DNA is still relaxed, okay? That's all about to change, however, as we head over into the mitotic phase, excuse me. Okay, so we're going to transition from interphase to this mitotic phase. So we're heading over here from G2 to the first part of the mitotic phase, which is mitosis. The first phase of mitosis is prophase. Okay, so we're doing this transition. The mitotic phase is shorter compared to interphase. The first part of the mitotic phase is mitosis, and there are steps basically as we go through prophase, metaphase, anaphase, and telophase, where we're going to show you how the cell separates this duplicated genetic material. Another way of saying that, how are we going to separate this sister from that sister into daughter cells, okay? So there's a lot of information. A lot of teachers focus on mitosis and sort of call it the whole process, but keep in mind the mitotic phase has two parts, mitosis and cytokinesis, and the mitotic phase in general is the shortest part of the mitotic cell cycle. Most of the time, the cell is in interphase. So let's discuss the first phase of mitosis, that being prophase, okay? And there are three key events that you should focus on when thinking about prophase of mitosis, okay? The first thing that happens, and you probably note a difference between how I'm drawing the sister chromatids in G2 versus how I'm drawing them in prophase. Okay, they are now condensed. The sister chromatids condense into smaller, now visible bundles of DNA. Prior to this in interphase, the DNA is actually not visible. Essentially, what this does is it makes the sister chromatids more manageable. They are easier to move around and eventually separate within the cell. This helps them also avoid being damaged because they're easier to move around. So condensation, the genetic material becoming more condensed, is the first step. You're also noting the difference between G2 and prophase, that the nucleus is starting to break down. This is a logical early step, as the nucleus basically represents an obstacle to dividing DNA. How could a sister chromatid separate and go to different ends of the cell if the phospholipid bilayer barrier was surrounding it? So the nucleus has to go, it breaks down, it fragments, and that's what we're showing here by the dashes, the black dashes there. Now the two centrosomes that we discussed earlier are going to migrate to opposite sides of the cell. So we're showing that right here. Okay, so the third part, centrosomes migrate, and eventually you're going to see these spindle microtubules basically push the two centrosomes away from each other so that we can spread to either side of the cell. So let's take a look at prophase now advanced over time, and you'll note that this centrosome over here, let's call it A, this centrosome over here, let's call it B, are now at opposite sides. And those centrosomes, their job is to send out spindle microtubules to make attachments to the sister chromatids. So both centrosomes send out spindles in order to grab the DNA, so to speak. Now, note that the fragments of DNA, they're still composed of two sister chromatids. Yes, they're condensed, they're easier to move around. We're going to transition from prophase over here to metaphase, okay? Now, as soon as the centrosomes have made contact with all of the sister chromatids in the cell, each centrosome will have contacted one side of that sister chromatid pair, as you see in this picture right here. And as both sides are pulling equally in a little game of tug-of-war inside the cell, the sister chromatid pairs will line up right along the middle of the cell in a place that we call the metaphase plate. Metaphase is a pretty easy phase of mitosis to identify, as all of the sister chromatids are lined up in the middle here along this metaphase plate. So let's transition from metaphase over here to anaphase. Okay, keep in mind that up to this point we haven't really done anything as far as separating genetic material. That's all about to change in anaphase. Anaphase is where the cell separates these sister chromatids. Consequently, as the sister chromatids are separated, the genetic material goes back to being called chromosomes. So all of these over here are now chromosomes. All these over here are now chromosomes as well. Okay? So basically, you're going to pull the chromosomes to either side of the cell, and they're being pulled by the centrosome. So in our example, we started with a 2n of 8. That means 8 chromosomes per cell. You can see here that the 2n is still going to be 8. Now we've actually separated those chromosomes. 1, 2, 3, 4, 5, 6, 7, and 8. Okay? Now to visualize this a little better, the enzyme that actually does the separating of the sister chromatids is cleverly called separase. So separase will come in here. Let me change this color so you can see it a little better. Separase is going to come in here and essentially separate the sisters here. Separate the sisters here. Separate the sisters here. Separate, 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 separates. And because the centrosomes are pulling with their spindle microtubules, now you're going to see basically the events of anaphase. The chromosomes are brought towards the centrosome. So basically, you're going to actually separate the chromosomes to either side of the cell. Now, as this is happening, the cell is also elongating. So you can see the cell is sort of stretching out here in the middle. That's because these spindle microtubules are also contacting each other and pushing away from each other in order to basically elongate as we get closer to the end of mitosis, okay? So basically we're preparing here for the events of telophase and cytokinesis. So let's advance from anaphase and show a picture of telophase and cytokinesis. Telophase is the last phase of mitosis. It essentially undoes everything that has been done to enable mitosis basically we're trying to get back to interphase. Our sister chromatids have been separated. Now each eventual daughter cell has its full complement of genetic material. We need to go back to interphase. Telophase basically does this by undoing all the steps of mitosis, okay? What's interesting too is the events of cytokinesis essentially cytokinesis, which physically separates the cell, the parent cell, into two daughter cells. It's going to happen towards the middle to end of telophase. Okay, so we have somewhat overlapping events here. We'll describe them together for you. But essentially what's going to happen here is the spindles, they need to be, the eraser is not working. The spindles need to be disconnected from the chromosomes. So basically this connection would be severed, okay? And essentially we need to put the nucleus back around our genetic material. The reason the mitotic phase is so short is that it's sort of a dangerous thing for the cell to divide, because your genetic material no longer is protected by the nucleus because we need to separate that duplicated genetic material. So we need to bring that nucleus back after disconnecting all these spindle microtubules over here, okay? So our nucleus returns, and you can also start to see that what was done in prophase, the condensing of your genetic material, is now going to be undone, and you can actually see the genetic material start to decondense. It's going to relax, okay? So the genetic material starts to relax. We've depicted that in the genetic material down here as well. As this is happening in telophase, cytokinesis is going to create a contractile ring here in the middle of this parent cell. It's going to squeeze the middle of that cell to physically separate the parent into two daughter cells. So we're going to get these two daughter cells after cytokinesis is complete, okay? This indentation here is called a cleavage furrow. So the indenting there physically helps the cell to separate through cytokinesis, and this is what we end up with. We have now two daughter cells that have the same amount of genetic material, a 2n equals 8, as that original parental cell, okay? So our one parent cells become two daughter cells. The genetic material has stayed the same. That's the job of the mitotic cell cycle, to take a 2n cell and make two 2n cells, okay? Now each of these daughter cells can then go on into interphase and start the process all over again. So we start with interphase. Both of these daughter cells will go through interphase. Their genetic material once again be loose. Okay, and the next step for them is to head to S phase. They want to head to the phase where they're synthesizing and duplicating DNA again. So that is a summary of the mitotic cell cycle with a focus on how the genetic material is changing. Is it loose? Is it condensed? Is it sort of a single chromosome, or is it a duplicated chromosome composed of two sister chromatids? I hope this helps to create a better mental picture of what's going on, not just in mitosis, but through the entire cell cycle, okay? The mitotic cell cycle encompasses interphase. It is not just mitosis. I also hope you're able to understand the quantitative aspects a little better. For my students on your test, you will run through some of the numbers, so to speak, about what is N versus 2N, what is diploid versus haploid. And I hope this video gives you a better idea of that, okay? We'll see you next time.